start, I would like to invite Marc Baggelaar from Utrecht University to give a presentation on exploring protein lipidation by advanced chemical proteomic strategies. Please give him a round of applause.
Yeah, thank you for the kind introduction and the opportunity to speak here today. So, as can be seen from my title, today I'm going to talk about protein lipidation. And I'm going to talk about protein lipidation in the context of the brain, because about 50% of the brain dry weight are lipids. It's thought that almost 40% of the synaptic proteins can be lipidated. Let's see how this works. So my lab works at the interface between chemistry and biology, and we use mass spectrometry proteomics to study proteins and protein post-translational modifications. So one part of our work is looking at the interaction of small molecules with proteins, and also the interaction of drugs with proteins. And what I'm going to talk about today is the lipidation of proteins, with a special focus on the brain. So we're developing methods to directly measure these lipids on proteins, which is quite challenging, and I will discuss that today. But at the same time, more established methods use chemical biology approaches with chemical tools to indirectly monitor protein lipidation. And we're working also on these strategies. But first, protein post-translational modification, which is another layer of complexity in our body. So we all know this central dogma. Oh, sorry. Where we go from about 20,000 genes, we have these various transcripts, about 100,000, and these can be translated into proteins. And these proteins can be post-translationally modified by a lot of different post-translational modifications, such as glycosylation, which is the attachment of sugars to proteins. It can be barcoded by acetylation or methylation or ubiquitination for degradation. But also fatty acids can be attached to proteins, which can change their function. And as a highlight of why these post-translational modifications are important, we have always this classical example of this caterpillar that turns into a butterfly. So they have exactly the same genome, but the phenotype is completely different because different proteins are expressed in these organisms. And then, another example is that we have here these different types of organisms that go from 4,000 to 20,000 protein encoding genes in these animals, and we see that it kind of levels off here at 20,000 protein encoding genes to build these complex organisms. However, when you add this layer of post-translational modifications, this becomes much more complex. And one additional advantage of these post-translational modifications is also that it's highly energy efficient. So you can just add a molecule to do chemistry on these proteins, and then you can change the function or the localization of this protein, instead of building a whole new protein. Let's see how this works. Yeah. So today we'll specifically focus on the post-translational modification protein lipidation, and there are many different types of lipidation. So we have this glycosylation, we can have N-acetylation on the N-terminus of proteins. We can have myristorylation, it's also on the N-terminus of proteins. Sorry, this was actually N-acetylation on lysine side chains. And we can have prenylation, which are these complex lipids. And for various of these protein lipidations, for these brighters, so these lipids are attached to proteins by specific enzymes. And for example, for myristorylation, there are already NMT inhibitors, which are these brighters of this lipidation, are in phase 2 clinical trials. And also for this farnesylation, there are also inhibitors for the proteins that attach these lipids to proteins in clinical trial. So also, in addition to its widespread nature of this protein lipidation, it's also highly druggable. And we have one special case here. This is S-palmitoylation, which has about 3,000 substrates in humans, at least that is thought. And this is the only lipid post-translational modification which is reversible. So there's a group of enzymes that attach it and a group of enzymes that remove it. And I will also specifically focus on this type of lipidation today. So here is this reversible cycle. So first these lipids need to be synthesized by the fatty acid synthetic machinery in our cells. Then these lipids can be loaded on these writers of palmitate, of which we have about 24 writers, and these attach these lipids to substrate proteins. And these lipids can again be removed by a series of about 10 enzymes are known to date that can remove these lipids from proteins. So you have this really dynamic cycle that can react to extracellular cues, for example. An additional advantage is that this tight regulation allows also to modulate it by inhibiting these writers or arrays of palmitate, and thereby we can also have an anchor to drug the system. So to recap, protein palmitylation or long-chain acetylation is very widespread. It's associated with inflammatory disorders, cancer and neurodegenerative diseases, and it's tightly regulated. So what are actually these consequences of these lipidation events on proteins? So once a protein is lipidated, it can do different things to a protein. So it can associate the protein to specific membrane subdomains. It can lead to conformational changes of these proteins. It can traffic them to vesicles to induce vesicle transport. And it can also mediate protein-protein interactions. And we can go even further in all these different functions of this protein lipidation. So it can multimerize proteins. It can do signal transduction and also open and close channels. So I think you get the point that throughout the years, many different functions of this protein lipidation have been identified. However, I've told you that it's very widespread and that it has important functions. However, this protein lipidation is highly understudied compared to other post-translational modifications like glycosylation or phosphorylation, at least in my view. And one of the reasons for this is that it's quite difficult to study because this protein lipidation it's highly hydrophobic, which makes it difficult to analyze. We don't have really enrichment strategies to enrich for these lipidated proteins or peptides we want to do proteomics. And this thioester is also relatively labile, so it can be removed during sample processing. And we have several strategies to analyze this protein lipidation. So one thing is what we can do is do acyl-biotin exchange, which I'll explain in a minute, where we swap the lipid for an enrichment handle. We can use this alkyne tag lipids to have a tag to enrich these lipidated proteins. And we can also overcome these limitations and do a detect-detection strategy. But I will first focus on this acyl-biotin exchange and how we use it to gain insight in the biology of protein lipidation. So one of the first areas that we looked in protein lipidation is the inflammatory response, and then specifically at macrophages, which are key cells in the innate immunity in response to pathogens or bacteria. And they have a special role because they can be both pro-inflammatory and anti-inflammatory, and they can switch between these different states. And then there you can also maybe appreciate that post-translational modifications could be highly useful to quickly change between these different states. And other reasons why protein lipidation could be important in this process, which is actually a highly simplified view of these macrophages. But lipidation is rapid, reversible, regulating acute polarization. We can maybe couple these inflammatory mediators directly to lipid modifications. We can couple metabolic reprogramming of the macrophages with specific proteins. And also we have this therapeutic relevance angle because we can target this protein lipidation and modify it by drugging the writers or erasers of this lipidation. So the strategy that we use in the lab to study this lipid post-translational modification is this isobiotin exchange, which I mentioned before, where we swap this lipid for an enrichment tag, which we can use to pull out these lipidated proteins. So what does it look like in a schematic view on a molecular level? So we have our lysate, so we have our protein pools, it's a complex proteome. We have several of these cysteines that can be lipidated through this thioester bond. Then first we can block all non-lipidated cysteines. And then we can after this blocking, we can hydrolyze the thioester, so releasing only the cysteines, so making only the cysteines available that were lipidated before. Then we can label these cysteines with the biotin tag, enrich them, digest them with trypsin, and then analyze them by mass spectrometry proteomics, and thereby identifying exactly the proteins, but also the sites that are actually lipidated. So we did it in THP1 macrophages, which is a monocyte cell line, and we can differentiate them towards M0 and M1 macrophages, and we can go from M0 to M1 with MPS and interferon gamma. And when we did this, we first wanted to check on the protein level, okay, if we do this induction, do we actually see an inflammatory phenotype? And indeed we did, so we saw several MHC class 2 antigens being significantly elevated, but also the JAK-STAT signaling was elevated, tryptophan metabolism. So we saw several markers of inflammation in these macrophages. So we thought, okay, we have a good system to actually look at protein lipidation in this inflammatory response of these macrophages. And when we did this lipidation analysis, this is quite a complex figure, but I'll talk you through. So every dot is a specific site which is lipidated, and here we compare the M0 versus the LPS interferon gamma stimulated macrophages. And I hope you can appreciate that we saw quite a large elevation of this lipidation in these M1 macrophages versus these M0 macrophages. And one other thing that we observed is that with this lipidation, that it was quite dominant in the transmembrane proteins versus the total proteome analysis. And when we were looking further at all these individual proteins which can be lipidated. One additional thing that we did in these macrophages, so we don't have very good inhibitors for this palmitate yet, but we have this 2-bromo palmitate, which is a non-specific inhibitor of protein S-palmitoylation, and we used it to see, okay, can we actually see a change in inflammation when we use this broad-spectrum inhibitor? And indeed, we did. We saw some inflammatory proteins going down in abundance when we treated them with 2-bromo palmitate. But interestingly, we did also a secretome analysis, and we saw a reduction in the secretion of several cytokines in these macrophages after inhibiting this palmitylation machinery. Then I wanted to switch to a slightly different setting, but it also has some overlaps. Multiple sclerosis is already explained quite thoroughly in the previous talks by Dan and Bart. And we also wanted to look at protein lipidation in multiple sclerosis because it has also a similar inflammatory phenotype, and we thought it could be interesting to look at protein lipidation in multiple sclerosis. So the questions that we asked ourselves is, can we measure long-chain acetylation in brain tissue? So we did this in an actual post-mortem human brain together with the group of Jeroen Bulgi at Hasselt University. And we wanted to see if there was an altered long-chain acetylation in MS lesions versus this normal appearing white matter, which Dan also explained. And the ultimate question is can we modulate long-chain acetylation? Could it be beneficial for patient outcome and multiple sclerosis? But it's a more longer-term goal. So for the first point, so can we actually measure this protein lipidation in these brain tissues? And the answer is yes. So here we see a comparison between this hydroxylamine positive and hydroxylamine negative treated samples. And this is really an indication that it's really thioester specific, so actually we're looking at protein lipidation. And we could identify in these brain samples over 2,000 lipidated proteins. And amongst these proteins that we identified to be lipidated are a lot of well-known lipidated proteins, so it's kind of a positive control. Then in the next stage, we actually wanted to compare the abundance of these lipidated proteins in lesions versus these normal appearing white matter. And there, just like in the THP1 macrophages, we saw an increase of inflammatory signature proteins that were being lipidated. We see that here. We see a whole series of proteins that are involved in inflammations which are upregulated in these MS lesions versus these normal appearing white matter. And we're now looking together with Jeroen in the role and, of all these different lipidated proteins. Then all these assays that I've mentioned so far use this acyl biotin exchange, where we remove this lipid from the proteins and then address it by a biotin, and in that way analyze protein lipidation. However, here we lose the information about the actual lipid that's attached, because we analyze these peptides and proteins after this lipid is removed from the protein. So what we're currently doing is we're developing methods to actually directly detect these lipidated proteins and these lipidated peptides, so we can actually see which lipids attached on the protein and at what site. So here's a sneak preview of recent data where we did this analysis. We can now do this for hundreds of proteins at the same time. And for example, here we see this direct detection data on PLP1, which is a key protein in the myelin sheets. It accounts for almost 50% of the protein contents in the myelin sheets. And there we see a large heterogeneity of lipidation events through these thioesters on these different sites of this protein. And this protein was known to be lipidated. However, it was not known which lipids were actually attached. And now we see a large heterogeneity of lipids attached to these proteins. So now we can also try to cross-correlate this with lipid metabolism, but also with the regulation by these different varieties of lipidation, which also maybe have different lipid specificities. And one additional feature, so we see this intracellular protein lipidation, but we see also several of these lipidation events on the extracellular space. And normally with these indirect methods, you would say this is likely a false positive. But with this direct detection method, we're quite confident that this is actually lipidation. So I've drawn this in this cartoon to be on this same membrane, but it could also anchor different myelin sheets together. But we still have to look into that. With that, I want to conclude. I hope I convinced you that protein lipidation is a widespread but understudied post-translational modification, and we still quite some work ahead of us, also within the ICNS consortium, where we want to apply this direct detection method. Protein lipidation plays a key role in the macrophage inflammatory response. We see upregulation of acetylation in these MS lesions versus normal appearing white matter. And I think that future lipid site-specific analysis of this protein lipidation will uncover new biology and also how this is actually regulated. With that, I want to thank especially Samiksha, Rose, and Andrea, who did a lot of this work that I presented, because all this optimization, all these essays, quite a lot of work and effort. And also Jeroen Begie and Jeroen Guns from Hasselt University for collaborating on this MS project, and also the funding agencies. And thank you for your attention.